Hello, I'm Alice Redgate, a member on Exeter's iGEM team for 2016. Welcome to today's episode of Desert Island Science. To watch more about the iGEM team's journey, you can find our channel by searching for Exeter iGEM 2016 on YouTube. Away today is Senior Lecturer at Newcastle University, Dr Jon Marles-Wright. An active member of the synthetic biology community, he was awarded a SynBio LEAP fellowship in 2015. His area of research expertise lies in structural biology, with a particular focus on the function of bacterial compartmentalization systems and their use as platforms for synthetic biology. So welcome, Dr Jon Marles-Wright.
Thanks for that introduction.
You've acted as an instructor for the University of Edinburgh iGEM team since 2014, and have been co-lead of the team since 2015. How did you first become involved?
Well, the Edinburgh iGEM team, I think it's it's the longest continuously running iGEM team in the UK. And it was run by Chris French and Alistair Elphick. So these are two fantastic, Chris is a microbiologist, Alistair is an engineer, and they came together and had some amazing projects, and they've been running the team for 10 years. And I think they got to the point where it's quite a lot of effort running an iGEM team, and they were wanting someone else to take over. So when Louise Horsfall and I started up in Edinburgh 3 or 4 years ago, they asked us if we wanted to be involved. So in the first year, we helped recruit the team and just helped advise the team. And then in the second year, we basically took over with Chris and Alistair stepping into a more advisory role.
What's your number one piece of advice for an iGEM team?
Get a really compelling project. I know that sounds a bit facile, but there's so many teams doing really lots of interesting things, and if you want to distinguish yourself from all the other teams, you have to have something that's really interesting and something that really engages with your local community. You see lots of projects that are really amazing but don't have any root in their community.
So what's your favourite project that you've seen so far?
I actually have to say we've been so concentrating on our own project this season that we haven't seen a lot of other projects. I think last year some of the projects where the William and Mary project that won was a brilliant piece of foundational synthetic biology. And it's nice to see teams coming up. You know, you've got your Imperial, your Brown, Stanford teams, really big teams doing amazing things year after year. But it's nice to see newer teams coming up and doing some really cool things.
So we're here today to find out about your views surrounding synthetic biology, but also to hear about the items you've picked to take with you on the desert island. So on the island, you already have a copy of the registry with access to all the synthetic biology parts, and a solar-powered bento lab that you're allowed to select one other book, one other piece of science equipment, one luxury item, and 2 songs. So can you tell us a bit about the first song?
So the first song, this is to remind me of last year's iGEM project. This is Feel Good Hit of the Summer by Queens of the Stone Age.
Why did you pick this song?
Because this is what the project was about last year. We were doing a drug sensor, and we were over the summer picking lots of songs that had drugs in them.
You mentioned that through an understanding of the basic biology and design principles of various metabolic compartments, you hope to use these as synthetic biology platforms. Can you tell us a bit more about this research?
One of the things we want to do with synthetic biology is get cells to make things for us. And a lot of the things we want, a lot of the natural products we want to make, a lot of the antibiotics we might want to make, are toxic to the cells. So the E. coli and the yeast we use as our chassis organisms, these compounds might be toxic to the cells. Now, a lot of most organisms kind of sidestep some of the problems with having toxic metabolism by putting it in organelles, separating it from the rest of the cell so it doesn't cause damage to the cell. And in bacteria, a number of different species have these little protein compartments that let them make use of different carbon sources that produce toxic intermediates. Now, one of the things we think would be really cool is if you took these little compartments and put metabolic pathways for the production of high volume chemicals of maybe toxic proteins inside the cells to separate it from the cell to increase your yield. But one of the things we don't know enough about how these things are made and exactly how they work, because you've got to get compounds in through the shell, you've got to get compounds out, you've got to get the proteins in and make them. So as a structural biologist, they're really fascinating because the way they put together from lots of different components and manage to fold up and get to the right place in the cell and do the biochemistry is amazing. And only through actually understanding how they work can we think about engineering them.
So how do you plan to engineer them?
So at the moment, I've got a PhD student working on putting things together. She's using Moclo assembly, but it's essentially breaking it down into parts, putting the parts together in different ways to see if we can recapitulate a functional system.
Can you tell us about the piece of science equipment you've selected?
So you've given us a bento box, which is brilliant, a bento lab, that's amazing. But I think the workhorse of iGEM, it's the multimode plate reader. You've got to have a plate reader, all those measurements of OD, measurements of fluorescence.
So a large part of our iGEM project is human practices, where we take our project beyond the lab. As part of this, we're planning to introduce a module in synthetic biology here at the University of Exeter. Given that you've previously taught courses on synthetic biology, what do you think are the most important principles for the subject, and how can we best engage students with this exciting new field?
In Edinburgh, we used to break it down into, we had two separate modules. We had a tools for synthetic biology and then applications. So we split out the kind of science bit and then what you can do with the science bit. And I think the important thing is, it's showing people that you need a lot of foundational tools from biology. Synthetic biology, it's not a discipline that sits on its own. It takes lots of bits and pieces from basic biology, from signal processing, from mechanical engineering, from systems engineering, chemistry, physics, and puts it together and does cool things. So we try to break it down. Here are the different scientific things that you could use as a synthetic biology. We teach about next-gen sequencing, about DNA assembly, about logic, and then put it into context. What do people make with their synthetic biology tools? How do they apply it? And in the Edinburgh course, we encourage lots of discussion. We give the students opportunities to present papers that they've seen that they think are good, to critique them, and really put it into the social context. What are the implications of doing these things? And we want our students to come out of our university, out of our courses, with an appreciation of how their science fits in society. And I think this is one of the key things in iGEM as well. You want to be responsible scientists, responsible synthetic biologists that are thoughtful. You know, if I make this construct, will it make my bacteria more toxic on a really basic level? And if I release these flies with a gene drive in them, will it wipe out other flies and destroy ecosystems? And just to have these discussions, and it's really fun actually, talking to students and saying, well, what happens if a gene drive goes wild?
So are both modules compulsory for your students?
In the synthetic biology programs, they are, yes.
Yeah.
But we get students from different programs, from the biotech program and biochemistry program, doing it just because it's so exciting doing synthetic biology now.
Can you tell us about the book you've chosen?
So you've given us the registry, but you need a lab manual. So I'm going to take the 3 or 4 volumes of Sandbrook and Russell. It's getting a bit outdated now, but if you want to set up a lab again, you've got to have that.
So what do you plan to make with your manual and bento lab?
Well, I guess the first thing, we're going to have to try and make some beer.
So let's have some more music. Can you tell us about the second song choice you've chosen?
So the second one is Race for the Prize by the Flaming Lips.
And why have you chosen this one?
This is just a great song about scientists. And scientists, we compete a lot, but really we'll work together.
The general public have a lot of concerns about the release of synthetic organisms into the environment. What are your thoughts on this, and how do you see the perception restricting the advancements of synthetic biology?
I think this is a difficult one. It's always difficult to say a general public. Who are these publics that are concerned about release of synthetic organisms? It's really hard to kind of pinpoint where people's particular concerns are and who is actually concerned. there's a lot of really vocal lobby groups.
When we went into a school a couple of weeks ago to give a talk on synthetic biology, and we asked the students what they thought synthetic biology meant, the first comment was design of humans, and the second was genetic engineering. What are your thoughts on that?
I think these associations are maybe harmful, and there's a sense that with the rebadging of genetic modification, genetic engineering as synthetic biology, it was trying to change the conversation from the conversation we had in the 90s and early 2000s about GM crops. It's worked to some extent. We're talking about it a bit more. We're being a bit more open and responsible and saying that there were some mistakes made in how we communicated with people. Let's try and rectify that. Let's do science in a more open way. And in general, science is becoming more open. We're saying we're not perfect. We'd like to have conversations. Let's see what people think. Do people want what we're doing? And at the bottom, if you're doing any kind of applied science, you have to ask yourself, do people want us to do this? Is it a useful thing to do? And solving world hunger, curing diseases, people clearly want that, but you can't force them to take new things that might be untested.
Would you be for the release of synthetic organisms into the world?
With many caveats.
What do you think needs to be done before that happens?
It's always a question of ecosystems. What's going to happen in the world ecosystem if you release an organism? Is it going to wipe out native species? We can't do that. We can't release organisms that are going to completely wipe out and destroy native ecosystems. So you have to be sensitive to what happens, and we don't really know. There are a lot of people interested in risk, but as scientists, we've often stepped back from that and said that's somebody else's problem. Mm-hmm. But now we think more about risk. What are the risks of releasing certain things into the wild? And with recombinant plants, with genetically modified plants, the risks are relatively low. But if you were going to destroy an ecosystem by taking out maybe a particular pathogen, a particular organism in an ecosystem, it's really hard to predict what happens. So I think you have to balance risk versus reward. And in a lot of cases, you probably don't need to release synthetic organisms.
Synthetic biology is a relatively new field of research, having only emerged within the last 2 decades. What do you think has been the most important breakthrough in synthetic biology so far, and what do you see for the future?
I think the really key, it's foundational for synthetic biology. It's next generation sequencing. Mm-hmm. Tons of sequencing data. We've got more parts to play with. Mm-hmm. And how cheap DNA synthesis is now. It's got to the point where if you can imagine it, you can build it, because DNA synthesis is so cheap. And those are really important breakthroughs that have just let us go wild with our imagination and do more exciting different things. And then we've built on that. Everything else has been a step on top of that. And I think what's coming in the future, it's automation. It's genome foundries. It's where everything we can imagine can be built. You put it together on your computer with a really neat little graphic interface. You push a button, and then a genome foundry, either in your lab or another lab somewhere else, it builds your device. It builds your organism, and then you get to test it. And that's really exciting because it takes out... Molecular biology is hard, mm-hmm. for no real good reason. And it's boring to do. And as scientists, we want to test hypotheses. We want to build things. The more and more cheaply we can do that and iterate our designs, the better. The more things you can test, the more you can try, the more you can build and learn, the safer things probably get.
So let's hear about the final item you've picked then, your luxury item.
It's a luxury item. You've got to take chocolate. Well, apart from the fact the bento lab is pretty luxury in a plate reader.
Any particular flavour?
Really nice dark chocolate.
So how do you think you're going to find living on the desert island?
I imagine it would be fun. I'm challenging it first, mm-hmm. but we'll sequence some genomes, we'll build some devices, and live in a lovely synthetic biology desert island.
Fantastic. So I'm going to cast you away now on our desert island. Thank you so much for talking to us today, Dr. Jon Marles-Wright.
All right, thank you.